All right, welcome to Chapter 9. This chapter is all about lipids and the roles that they play in biological membranes. So in this chapter, we're going to start in this particular lecture talking all about lipid classification. And among the different classes of lipids, we're going to discuss our fatty acids, triacylglycerols, glycerophospholipids, sphingolipids, and steroids. In future lectures, we will be discussing lipid bilayers and membrane proteins. As well as membrane structure and assembly, which are really, really important roles within a cell that lipids are playing. So first of all, what is a lipid? Let's all get that definition clear in our minds before we start discussing them. So lipids are classified as molecules that are soluble in organic solvents, meaning nonpolar solvents. And so commonly we think of lipids as fats and oils. Lipids, unlike a lot of the structures we've discussed so far, things like proteins and sugars and nucleotides, those three main things that we've discussed before are all able to be formed into long polymers. Lipids don't form polymers, but they do tend to form really large assemblies and interact with each other, but not through necessarily covalent bonding. So, for example, in a cell membrane, we have a whole lot of lipids that are all interacting with one another, but they're not polymeric. They haven't formed covalent bonds with one another. And aside from their role in cell membranes, lipids can have a wide variety in their structures, and as we've talked about before, variations in structure can lead to a lot of different variations in function. So, first let's talk about fatty acids, which are components of many lipids. So fatty acids are defined as a carboxylic acid and a long hydrocarbon chain. So if we look at our example here on the right, we have stearic acid. We have our carboxylic acid here at our carbon label number 1. And the rest of the carbons number 2 through 18, are this long hydrocarbon chain. That's just carbons and hydrogens. And biologically, when we look at fatty acids, they tend to be in even numbers, generally from 14 to 20 carbons, with 16 or 18 carbons being the most common ones that we see, particularly in biological membranes. And part of the reason for this is that when lipids are being synthesized biologically, they tend to be in pairs of 2 carbons at a time. So that's why we end up in these even numbers of carbons when we're looking at fatty acid molecules. When we're looking at fatty acids, something else we need to really consider that's very important is saturation. So saturation can affect the shape that this fatty acid will take on. So in this case, our stearic acid, the original fatty acid that we looked at, it is saturated. So it contains all single bonds in that long hydrocarbon tail. Of course, we don't consider the carboxylic acid part of it when we're talking about this, but just the actual hydrocarbon part, it is all single bonds. In contrast to this, we have a couple of other types. So we have, depending on how unsaturated the fatty acid is. So our next example here is oleic acid. Oleic acid is called monounsaturated because it has one double bond, and that's seen here in red. So if we number our carbons, starting at number 1 being the carboxylic acid carbon, and number all the way down, it would help if I could write numbers in the right order. We have our carbon number 9, where our double bond is, and this will come into play in a couple of slides when we start talking about how to name our hydrocarbons. So our double bond begins at the 9th carbon from the carboxylic acid, which is carbon number 1, in oleic acid. There are also fatty acids that have more than one double bond, and these are called polyunsaturated fatty acids. So we have 2 examples in this slide. First, we have linoleic acid. This one has 2 double bonds. The first one starts on carbon number 9. The second one starts on carbon number 12. We also have alpha-linolenic acid. Alpha-linolenic acid has 3 double bonds. So it has one starting at carbon 9, carbon 12, and carbon 15. And so both linoleic and alpha-linolenic acid are called polyunsaturated. So when we start to think about saturation, that starts to affect the properties of the fatty acid in terms of how it interacts with other fatty acids. So when we're thinking about saturated fatty acids, again, this is the one that has all single bonds, like our stearic acid here. It is at its most stable when it's in this relatively linear structure that we see pictured here. So remember that all of these single bonds that we see connecting in this hydrocarbon tail all have free rotation about those single bonds. But when all the bonds are at these particular angles where the structure is relatively linear, that allows multiple saturated fatty acids like this to all pack together very tightly. And this helps them to better exclude water, which helps them become more stable. And so the longer these hydrocarbon tails are, the higher melting point they have, meaning they're a long hydrocarbon chain means it's even more stable, it can pack very tightly, and so it doesn't want to melt. It has a high melting point. This is in contrast to our unsaturated fatty acids, where the double bond, in particular this cis double bond that we see here, introduces a bend into that hydrocarbon chain. It's the way it's drawn in this picture, it still looks fairly linear here, but you'll see in some future slides in more space-filling diagrams, you actually get a pretty severe angle. And this angle prevents those hydrocarbon tails from packing together so tightly, the way that saturated fatty acids would. And so unsaturated fatty acids tend to have lower melting points because they are a little less able to pack tightly and a little bit more easy to break apart, and so they're more easy to melt even at lower temperatures. So let's start talking, how do we talk about unsaturation? How can we describe where the location of the unsaturation is? So we have two sort of options, right? We can number from the carboxyl group, and that's what we had started to do on the previous slide. Or you can start numbering from the other end, and we'll get into that in just a moment. So first, let's consider our stearic acid. This is the fatty acid that's pictured on the left here, the completely saturated one. Its common name is stearic acid, but as an 18-carbon fatty acid, it also has a systematic name, which is octadecanoic acid. We can break this down for octa meaning 8, deca meaning 10, so 8 plus 10, that's 18. Oic acid referring to the fact that it's a carboxylic acid. And it has no double bonds, right? It's completely saturated. So let's compare this to oleic acid, which is pictured right next to it over here. So oleic acid still has 18 total carbons in it, right? So we still have that octadeca in the name for 18, but we have one double bond. So instead of anoic acid, like we had in stearic acid, now we have enoic acid. So we have one single bond, and it's got 18 carbons. So now we also have to describe it by saying where that double bond is. So for our systematic name, we start labeling from the carboxyl end. And if we start numbering those carbons, like we were talking about on the previous slide, that double bond starts at the 9th carbon over here from the carboxyl end. And so we can call this by a more systematic name. Instead of oleic acid, we can call it 9-octadecanoic acid. But for any unsaturated fatty acid, we can also start counting carbons from the other end. And this is called the omega system of naming, because instead of starting from the beginning, we start from the end, omega being the last letter of the Greek alphabet. And omega positions for double bonds are more important from a health perspective. You may have heard, eat your omega-6 fatty acids. This is what we're talking about here. We're talking about the location of a double bond in a fatty acid. So if we're talking about oleic acid, if you want to figure out the numbering for the omega system, we would start numbering carbons from the other end. So this carbon here that says 18, it's the omega carbon. So we would actually call this carbon number 1 if we're talking about the omega system. So if we start numbering double bonds coming up this way from the omega end, we still have our double bond starting at the 9th carbon. And this is just because this particular fatty acid has 18 total carbons. So if our double bond is right in the middle, it's 9 away from both ends, right? It's a little bit different if you start looking at some of these other polyunsaturated fatty acids that we were talking about before, like linoleic acid. So let's take that as our next example. Linoleic acid has 2 double bonds, so its suffix for the systematic name would be a dienoic acid. And those double bonds are located 9 and 12 carbons from the carboxyl group. So in the systematic name, we would call it 9,12-octadecadienoic acid. But for the omega system, like I said, we start from the other end. So if we wanted to count this for the omega system, our first double bond is located at the 6th carbon from the omega end, and so it is an omega-6 fatty acid. And when we're using the omega system, we tend to just stop after the first double bond that we encounter. So for the omega system, we can ignore this second double bond over here. So our fourth example now is our alpha-linolenic acid. So alpha-linolenic acid has 3 double bonds located 9, 12, and 15 carbons away from that carboxyl group. So its systematic name would be 9, 12, 15 octadeca, and now it's trienoic acid. And in terms of the omega system, if we start counting from the end, our first double bond starts 3 carbons in, so it's an omega-3 fatty acid. So let's talk a little bit more about those double bonds. So if you recall, double bonds can either have a cis or a trans configuration, depending on where the R groups are and where the hydrogens are. So for most of the fatty acids that we see in nature and in our cells, we most frequently see cis double bonds. And like I mentioned before, that double bond introduces essentially a kink or a bend into the structure of that long hydrocarbon tail, which you can see in this picture here. And this is in contrast to this picture up here in blue, which shows a completely saturated fatty acid. With the pink background, we have an unsaturated fatty acid with a cis double bond. And I believe I also mentioned this before, but because of this bend in the hydrocarbon tail, these fatty acids are not able to pack as efficiently. And so this means the more double bonds that we have in any one fatty acid tail, the lower the total melting point is going to be, because the less it's not going to be able to pack as tightly, so it's going to be easier to pull them apart even at lower temperatures. And so this is in contrast to trans double bonds, which we see pictured down here with our green background. And so trans double bonds, we don't often see them in nature, although we will see one example of them in our slides during this lecture. We most frequently see them as a byproduct of manufacturing when oils are being hydrogenated to make solids, to make solid lipids. And so in a trans fatty acid, the structure remains linear, right? So in our cis double bond, we have this very obvious bend in our fatty acid. In our trans double bond, the shape of it still overall looks a lot like the saturated one, right? It has that same long linear shape. And so this means it's able to pack pretty closely, pretty well, very similarly to the way that a saturated fatty acid can. But in reality, it doesn't behave completely like a completely saturated fatty acid, because you have some limitations in rotation, that kind of thing, in a trans fatty acid because of that double bond. And so ultimately, the packing and the melting point are close to saturated fatty acids, but they're really somewhere in between saturated and the cis unsaturated ones. That is supposed to say cis. I apologize. All right. So now let's take a moment and think about all that stuff that we just took in about fatty acids and degrees of unsaturation and length of tail. And let's try to rank each of these groups, group 1, 2, and 3, of different fatty acids from lowest to highest melting point. Before you begin this exercise, I just want to let you know that I used a slightly different naming convention here when labeling these 3 groups. So here, to describe each of these fatty acids, we have in parentheses 2 numbers separated by a colon. The first number in this set in the parentheses is the number of carbons. The second number after the colon is the number of double bonds. And if it doesn't say, you can usually assume that that double bond is a cis double bond. So that should be enough information to get you started on this group of questions. So I will invite you to pause the slides here and take a few minutes for yourself to try to solve each of these problems. And we'll rejoin in a few seconds. All right, so let's start discussing the melting points of each of these groups. So let's start with group 1. So as you can see, oleic acid, linoleic acid, and alpha-linolenic acid, excuse me for this typo, linolenic, they each have the same number of carbons, right? If you look in the parentheses, they each start with an 18. So these are each 18 carbon fatty acids, so we can't use the fatty acid chain length to help us determine the melting point. The only difference between them is the number of double bonds. And so it might help you to think about the ability to pack, how loosely or tightly these can pack, how much freedom of movement each of these fatty acids has. So if we're talking about oleic acid, the first one on our list, it has just one double bond. And so of these three, it has relatively the best ability to pack because it only has one double bond that is introducing a funny shape to it. And so this one is going to have the highest melting point. So it's going to be number 3 on our list because we're labeling from lowest to highest, right? If we want the fatty acid with the lowest melting point, we want the fatty acid that has the least ability to pack tightly. So that's going to be the one with the most double bonds, which is alpha-linolenic acid, number 1. Linoleic acid is going to fall somewhere in between those two. So now let's consider group 2. Group 2 has 3 different fatty acids, myristic acid, palmitic acid, and stearic acid. The difference between them that we can see based on that information in the parentheses is how many carbons each of those fatty acids has. And we can see because of the 0 after all of those colons that they're all completely saturated. They have no double bonds. So if we're talking about melting point, like we discussed before, the longer the fatty acid chain or tail, the higher the melting point because the more tightly it wants to pack. So again, our highest melting point, our third one, is going to be that longest fatty acid. And so in this group, that's our stearic acid. Our lowest melting point, that is the one with the shortest fatty acid hydrocarbon chain that has the least ability to pack, sort of the most freedom of movement, is going to be our myristic acid. And our palmitic acid will be somewhere in between. And so last, let's now consider our group 3. Group 3 has 3 different fatty acids. They each have the same number of carbons, right? So stearic acid, oleic acid, and aleatic acid each have 18 carbons. And the only difference between them, yet again, is the number of double bonds. So we have stearic acid, which is unsaturated and no double bonds. Oleic acid has one double bond that's in the cis configuration, and aleatic acid has this double bond in the trans configuration. So if we're talking about the lowest melting point, we're going to want the one that can pack the least well, right? That's the one with the cis double bond. That's our oleic acid. Our highest melting point is our completely saturated fatty acid, right? So that's going to pack the most efficiently. Highest melting point. And then somewhere in the middle is where we are going to put our trans double bond. And so let's see how we did. We have the melting points now for each of these fatty acids. So we can see now for group 1, our lowest melting point is our alpha-linolenic acid, very cold. Our higher melting point is oleic acid. But as you can see, all of these are below room temperature. So these 3 different types of fatty acids are all going to essentially be oils at room temperature. In group 2, we have our saturated fatty acids, right? And again, we see this trend in increasing melting point, but all of these melting points overall are a lot above room temperature, right? So all these are going to be solids at room temperature. They are not able to melt unless you heat them up a little bit. And then in group 3, we have this mixture, right? So oleic acid is going to be a liquid at room temp, and the other 2 are going to be solids at room temp. And so the effect of double bond is separate and distinct from the effect of the length of the hydrocarbon chain. So it's very easy to rank them when they're grouped like this, where there is only a difference in the length or there is only a difference in the number of double bonds or the type of double bond. It's much more tricky if you have a combination of differences in both length and double bonds. I would not ask you to do that, for example, on an exam. All right, so let's continue on. So now we've defined fatty acids. Another word for fatty acid is an acyl group. So it has a carboxylic acid on one end and a long hydrocarbon chain. This is referred to as an acyl group. And so when we talk about triacylglycerols, triacyl tells us there are 3 acyl groups or 3 fatty acids, and they're all on glycerol. So first of all, what is glycerol? So we have a picture of glycerol over here on the right. Glycerol is a 3-carbon chain, and each of those carbons has an alcohol on it, a hydroxyl group. So you can think of each of those hydroxyl groups as sort of potential attachment points for other groups, often through an ether linkage, but not always. And so when we're talking about a triacylglycerol, we do see that ether linkage. So we have our sort of original glycerol backbone here with our carbons numbered 1, 2, and 3. We have 3 different fatty acids attached through an ether linkage from the carboxyl end, and that is what forms our triacylglycerol. And the 3 fatty acids can each be the same, or they can each be different. It depends, or it doesn't matter. You see a variety. So what is the function of triacylglycerols? In plants and animals, they are a really common means for energy storage. And they're very efficient compared to an energy storage medium such as glycogen. So first of all, the carbons in triacylglycerols are more reduced. So if we think back to a few lectures ago, we talked about how ultimately during cellular respiration, we are ultimately oxidizing carbon such that our end result is carbon dioxide, right? CO2. This is the most oxidized. And so if you start from a carbon in, let's say, a sugar, most sugars, the carbons already have hydroxyl groups on them. So they're already sort of partially oxidized. But if you're thinking about a fatty acid, most of those carbons have all hydrogens on them. And in fact, the most fully reduced carbon you can think of is methane, right? Because it's a carbon with all hydrogens on it. This is the most reduced type of carbon. So if you start out with carbons that are more reduced, you have the more potential for oxidation events on those carbons. And so essentially you have more sort of energy available to you sort of in there because you're starting from a more reduced state. So you have more distance to go before it gets fully oxidized. All that is to say, triacylglycerols are fairly reduced and compared to a molecule like glycogen, and so they are a more efficient way to store energy. And so triacylglycerols, when we store them, they are stored in an anhydrous form where we exclude water in cells called adipocytes. And they, triacylglycerols, gram per gram, compared to glycogen, contain about 6 times as much energy. If you think about, again, that conversion process using oxidation. And so if you want to think about that in terms of how much fuel that can provide you, the triacylglycerols we have stored in our body would have the potential to sustain us with enough energy for about 2 to 3 months, compared to our glycogen stores, which only have realistically enough energy in them to supply us for about 1 day. So let's now switch gears a little bit and think about triacylglycerols as a form of storage that we see in plants and compare them to what we see in animals like us. So there are 3 different main categories where we see differences here. So the first one is whether these triacylglycerols contain solid fats or oils. So in plants, generally they tend to have oils, which are often more unsaturated. In animals, the triacylglycerols tend to be more solid fats, which tends to mean we have more saturated fatty acids, which I just then again gave away our next clue here, saturated versus unsaturated. So in plants, our oils are unsaturated. In animals, our solid fats are saturated. And so what does that mean for whether they're liquid or solid at room temperature? Hopefully by now you've caught on with the scheme here. And so we have our oils, they're unsaturated, they're liquids at room temp. Our fats, our solid fats, they're saturated, they're solids at room temp. All right, so let's talk a little bit about this example of a triacylglycerol. So the name of this triacylglycerol is written on the bottom. So the naming convention here, we can start by looking at our original glycerol, which we have labeled here in black. And the carbons on the glycerol are labeled carbon number 1, 2, and 3. Each one of those carbons has a different fatty acid tail attached to it. So attached to carbon number 1, we have this palmitoyl group. So palmitoyl, we know, has 16 carbons in it, which we can see numbered down here. And we also have this double bond here at position 9, and so this particular group with 16 carbons and a double bond at the 9th carbon is called palmitoyl. On the 2 carbon of our glycerol, in the middle here, labeled in blue, we have our linoleoyl. So this is an 18 carbon fatty acid with 2 double bonds, just like we saw in our linoleic acid. And in our 3rd position on the glycerol, which is carbon number 3, and it's shown in green here, we have our completely saturated fatty acid, and this is our sterol. So again, it's based on stearic acid. It's got 18 carbons, no double bonds. So overall, you could call this whole structure 1 palmitoyl, 2 linoleoyl, 3 sterol glycerol, which is a bit of a mouthful. And so if you think about this as more of a 3-dimensional structure, this green fatty acid tail, which is completely saturated, would be relatively linear looking. But if you made a space filling diagram, the fatty acids at positions 2 and 3 would have additional bends or kinks in them because of those double bonds. And so that would make this take up more space than if, for example, all 3 of the fatty acid tails were saturated. So let's start to talk about some sort of variations on a theme here. So next we're going to talk about glycerophospholipids, which are the major component of biological membranes. So they have a lot in common structurally with triacylglycerols, right? They have a central glycerol. Remember, glycerol has 3 carbons, each of which can be attached to something else. And so just like in triacylglycerol, 2 of those carbons are attached to a fatty acid. In triacylglycerol, the 3rd carbon on the glycerol is also attached to a fatty acid, but in the case of glycerophospholipids, like the name suggests, the 3rd carbon in the glycerol is attached to a phosphate and alcohol group, which is sometimes called the head group. And so phosphate groups, as we know, are charged and negative. Alcohol groups are polar. So this whole end of the glycerophospholipid molecule is polar. And it's also hydrophilic. And that's very different from our fatty acid end of the molecule, right? So this is what we frequently will refer to as the tails, or the tail, because fatty acids have those long hydrocarbon chains. They kind of look like tails. I think I've been saying tail throughout this lecture, very common nomenclature. And these fatty acid tails, as we've been discussing, are completely hydrocarbon based, so they are very nonpolar. And that means they are also very hydrophobic. And those are the components of glycerophospholipids, and we'll talk a little bit more about them here. But first, we're going to discuss what are the different potential components of that head group, right? So I should go back and say this phosphate group is always there as part of the glycerophospholipid, but what is labeled here as alcohol, this is variable, right? And so it's referred to as X on the next table, which we're going to look at in just a moment. So the X is shown in this middle column, right? That's that sort of end of that head group, which is attached to a phosphate, which is attached to a glycerol, which is attached to 2 fatty acid tails. So the simplest one is just if there is a water there or nothing, and this phospholipid would be called phosphatidic acid, and the other types of glycerophospholipids are named sort of based off of this name. So if we have an ethanolamine there as that alcohol sort of group, we would call this phosphatidyl ethanolamine. If we have choline there, it's phosphatidylcholine, etc. You can go down this list. You can have sugars there like inositol. You could even have another phosphate containing group like we see here on the bottom, this phosphatidylglycerol. You can have another glycerol there. You can have all kinds of stuff. So, and we see a lot of these are different components of the cell membrane. This last one in particular, cardiolipin, is an important and unique component of the inner mitochondrial membrane. But you will definitely encounter these again when you start looking at components of cell membranes. So let's look at this particular example and start breaking it down. So we have our central glycerol, right? Remember, this is a glycerophospholipid. It's all in the name, right? So start with the glycerol. We have two fatty acid tails. Carbon number 1 of the glycerol has this fatty acid tail, which we know from our name here is sterol, right? Which is our 18 carbon saturated fatty acid tail. Our second carbon in the glycerol is linked to an oleic acid, or what is written here as oleol. And then our third carbon in our glycerol is where our phosphate-containing head group is attached. So in blue here we have our phosphate group, and then our alcoholic head group attached to there is choline. So altogether we would call this molecule 1-sterol, 2-oleol, 3-phosphatidylcholine. All right, and if we look at what that would actually look like with a space-filling diagram, it has a number of different important components. So we have our polar head group sort of on the top here. This contains our negatively charged phosphate group as well as our positively charged choline. And then attached on the other end of the glycerol is our fatty acid tail. So the tail end is nonpolar. And again, this space-filling diagram makes it really easy to see with our oleic acid, this very distinct bend or kink in the hydrocarbon chain of our fatty acid as compared to our long, relatively linear stearic acid component. And so if you can imagine if you had a bunch of these and you wanted to arrange them all next to each other, you might have a problem where they're bumping into each other in space because of the bend introduced by that cis double bond. And once again, the top part over here on this diagram is the head, which is polar and often charged, and then we have the tails, which are nonpolar. And so this makes the tails want to interact with other nonpolar things, and the heads want to interact with other polar things, or water. So before we continue further with this, let's talk a little bit more about how lipid bonds can be broken. And this is done using enzymes that are called lipases. And so if you look at this diagram here, this sort of space-filling structure here is a glycerophospholipid, and it's getting cleaved by an enzyme, which is a protein called a lipase. And you can see the different amino acid structures and side chains on the left of this figure, kind of surrounding the polar head group part of this glycerophospholipid. And you can see all in the parts of the lipase that are interacting with the lipid, nonpolar part of the lipid, you can see these bulky, nonpolar kind of hydrocarbon side chains, and those will help to stabilize the nonpolar parts of the lipid. So once you lyse a lipid bond, you have what we call a lysophospholipid. Lyso because it's been cut, right? So lysophospholipids have distinct functions compared to whole glycerophospholipids. Lysophospholipids tend to act as detergents, which means they're able to disrupt membranes or to lyse cells. And so if you look at our diagram on the bottom here, we have our phospholipase. This is a type of lipase, right? It hydrolyzes lipid bonds, and our product after that cutting is a lysophospholipid. And so we can see here's our central glycerol, carbons 1 and 3. Carbon 1 has a fatty acid tail. Carbon number 3 has the polar head group attached. Carbon number 2, that fatty acid has been cut and replaced by hydrogen. And we have the free fatty acid shown here as a product of this reaction with phospholipase A2, which cuts right here, if this is our original glycerophospholipid. There are also a number of different classes of phospholipases that all have different slightly different names. So phospholipase A1 and A2 cut that ether linkage between the glycerol and the fatty acid, either at carbon 1 or carbon 2 of that glycerol, based on the name. Phospholipase C and D cut somewhere on that head group, either before or after the phosphate. And you'll see as we learn about signaling a while from now, some of these phospholipases might come back, as well as some of the products of the cutting process from some of these phospholipases. They can act as signaling molecules that can ultimately help give a cell directions for how to behave or function. So let's talk about some other components of biological membranes, which are also lipids, but they are not based on a glycerol backbone like the other lipids we've discussed so far. So one that we want to discuss now are sphingolipids. So sphingolipids are based on sphingosine. Sphingosine is an 18-carbon amino alcohol. So let's look at what that means. So if you count up all the carbons in this molecule, there are 18 of them. I won't go through the trouble with you of that right now, but you can count them yourself, or just trust me, there are 18 of them. We have this amine group here, making it amino, and we have 2 alcohol components. And so the way it's drawn, this part looks a little bit like that 3-carbon glycerol with the alcohol groups on it. But it's a little bit different, right? That central carbon in particular has an amino group that we have circled here, and that this carbon that's attached to that rest of the carbon chain that's part of the sphingosine contains this double bond right here, and this is a trans double bond. And so this part, if you're not looking carefully, it looks maybe a little bit like an ether linkage to a glycerol would look, but it is not, right? There's no ether linkage. There is, in fact, just a trans double bond, and further, this is all just one single molecule. It's not a glycerol with different things attached to it. So that's sphingosine. And then we can have a bunch of derivatives of sphingosine as well. So one of those derivatives is called ceramide. Ceramide is an N-acyl fatty acid derivative of sphingosine. So we can see on the bottom of the slide is ceramide. The original structure shown in black is our sphingosine with our 18 carbons, our trans double bond over here, our amino group over here, and our alcohols up top. When we add on a fatty acid residue to that nitrogen, which is why it's called an N-acyl, because we have the acyl group attached to a nitrogen, that's what makes it a ceramide. So adding on to there, we have sphingomyelins. Sphingomyelins are ceramides with either a phosphocholine or a phosphoethanolamine head group. So again, let's look at what that means. So in this figure shown in black, we have our original sphingosine, that 18-carbon amino alcohol. In red, we have a palmitate residue, that's our N-acyl attachment, making it a ceramide. And then in addition, on this carbon at the end, we have, shown in blue, our phosphocholine head group. And that head group is what makes it a sphingomyelin. Sphingomyelins are really important components of our cell membranes. They make up about 10 to 20% of our membrane lipids. They have really similar conformation and polarity to glycerol phospholipids, which are also very important components of our cell membranes. And they all sort of live together in the cell membranes because they're relatively similar to one another. Sphingomyelins in particular are very abundant in the myelin sheath. So going on with that brain idea, right, with myelin sheaths, we can now consider cerebrosides. Cerebrosides are a little bit like ceramides, except they have a single sugar head group. So this picture is a little bit different than the other one, so let's break it apart a little bit. So we have here our central sort of 3 carbons that we talked about before, kind of looks almost like a glycerol, but it's not. We have our amino group that has been acylated with a fatty acid tail. That's what makes it a ceramide, in addition to this trans double bond linkage, as well as this other alcohol component. And so that's our sphingosine/ceramide backbone. And then instead of a A head group, like we saw with sphingomyelin, that's made of phosphocholine or phosphoethanolamine. For cerebrosides, we have a sugar, which I am circling right now as the head group. And in the brain, the primary lipid that makes up a lot of membranes within the brain, we have galactosylceramide. So the sugar would be galactose. So moving on from there, we now have gangliosides. Gangliosides are similar but different. We still have a ceramide, right? So we have our original sphingosine that's shown in black here, plus our N-acyl attachment shown in green, helping to make it a ceramide. And then our head group, instead of being a single sugar, is now an oligosaccharide, which is shown here in blue and red. And so gangliosides are really important as cell surface receptors, and they play a really important role in cell-to-cell recognition. So before we move on from there, all of these different lipids that we just went over are all different sphingolipids, right? They're all based on sphingosine, and then once we have sphingosine, a lot of them are actually based on ceramide, which is sphingosine with one fatty acid. Then our other ones, our sphingomyelins, cerebrosides, and gangliosides, are all ceramide, but with different sort of head groups. All right, so now that we've gone over those sort of components that are the main components of a lot of biological membranes, we're going to switch gears a little bit and talk about another really important class of lipids that we do actually also see a lot in our cell membranes, but that has a very different structure. These are steroids. So steroids are cyclopentanoperhydrophenanthrene derivatives. That is a very long word that refers to this four-membered ring structure that I will now just call a steroid backbone. One really common steroid that we probably are all relatively familiar with is called cholesterol, which we see here pictured on the bottom. Cholesterol is a type of sterol, and a sterol is a steroid that has a hydroxyl group. So as we can see, cholesterol has this steroid backbone that matches our ring structure at the top, and in addition it has one hydroxyl group, making it a sterol. The rest of cholesterol that we see here has a number of other different substituents at different locations on the ring backbone, but overall the rest of it is very nonpolar, mostly just hydrocarbon based, and this region of it is slightly polar in comparison, where we have our hydroxyl group. And cholesterol is in animals, it is a major component of our plasma membrane. It's up to 30 to 40% of the plasma membranes of our cells. And and cholesterol has a little bit of this sort of amphipathic nature where it's a little bit polar on one end, but it's majority nonpolar. And this, especially this nonpolar region of it, helps it to sort of insert itself into our cell membranes, kind of in between a lot of those fatty acid tails, where it can affect some of the properties of the fluidity of the cell membrane. So let's talk a little bit now more about some other types of molecules that also utilize that steroid backbone that have a very different function. And what I'm referring to now is the steroid hormones. So steroid hormones have that same steroid backbone or ring structure, but steroid hormones act as signaling molecules that can send messages between different tissues, different organs. Steroid hormones are usually transported by other proteins to their target tissue, where they can then have an effect on the cells in that target tissue. Some steroid hormones can be further classified into a number of different categories. And so the first one we're going to mention here are the glucocorticoids. And one example that we see over here is cortisol or hydrocortisone. And you can see here it still has that four-ring structure with a number of different substituents on it. And so what just a few things to know about glucocorticoids. They are synthesized by the adrenal cortex, and they are involved in all kinds of different signaling, including for carbohydrate, protein, and lipid metabolism, as well as inflammation and the stress response. Another type of steroid hormone is the mineralocorticoids, and the example we see here is aldosterone. Mineralocorticoids are also synthesized by the adrenal cortex, and they are involved in the excretion of salt and water by the kidneys, which makes them really important for keeping the correct balance inside of our bodies. Another type of steroid hormone are androgens and estrogens, or sex hormones. And we have two examples here. We have testosterone, which is an androgen, and beta-estradiol, which is an estrogen. And looking at all four of these structures now, you can all see how similar their backbones are, but how different the other components that have been added on are. So androgens and estrogens are synthesized by the testes and the ovaries, and they are really important for our sexual development and function. So keeping on this theme of steroids, now we're actually going to talk about vitamin D, which is also derived from steroids. So you can see this steroid backbone from this vitamin D precursor, and after some rearrangements and breaking and movement of bonds that is both caused by some enzymes, as well as you need the UV radiation for some of it, which is why we talk about sunlight being so important for making vitamin D. You can make, there are several different forms of vitamin D that you can see on this slide. So we have shown over here vitamin D3 and D2. But what you essentially need to know here is that vitamin D is just another derivative of a steroid. And so vitamin D is one type of vitamin that is fat soluble. There are also other vitamins that are fat soluble. These include vitamins A, D, E, and K. And these little blurbs have a little bit of information about each of these fat-soluble vitamins. So vitamin D is the one that's derived from steroids, but all four of them are fat-soluble. They're hydrophobic, right? Vitamin A is really important for eyesight and the immune system, as well as for skin. Vitamin E is really important as an antioxidant that helps prevent damage by free radicals. Vitamin D, as we were just talking about, is really important for bone health as well as your immune system. And vitamin K is really important for blood clotting as well as in bone health. So with that, you are now ready to answer the Chapter 9 Lipid Classification practice questions.